Welcome to this instructional video to finish out Chapter 5. So we're going to skip the page on clavulanic acid and we're going to go right into a topic called sarin gas. This is an example of irreversible inhibition. Well, here's the story. There's a molecule called acetylcholine. We've mentioned this molecule before briefly. It's a neurotransmitter. It is released in the synaptic clefts and it propagates electrical signals. But we need to break it down. And so there's an enzyme called acetylcholine esterase, and this esterase, hey, it breaks the ester bond in acetylcholine. And this turns off kind of the nerve propagation signal. Well, what we know is that many years ago, there was a very powerful poison nerve gas developed called sarin. And you can kind of look at the structure of sarin right here. It's a small molecule, but it is very, very deadly. It has a lethal dose, an LD50, equal to 550 micrograms per kilogram of body weight. That is in a rat, not in humans. But I think we can safely say that sarin is highly, highly toxic. Why is it toxic? It turns out that sarin is an irreversible inhibitor of acetylcholine esterase. If you look in the active site of acetylcholine esterase, let's abbreviate that ACHE. Here's the active site. There is one particular serine residue in acetylcholine esterase, and it can actually attack the sarin molecule if the sarin molecule comes into the active site. Remember, with irreversible inhibition, the irreversible inhibitor goes into the active site and it will chemically react with the enzyme, a side chain of an amino acid in an enzyme. I don't want to get too deep into the mechanism of this, but what we know is that serine can act as it's a pretty weak nucleophile, but guess what? The phosphorus atom of sarin gas is highly electrophilic. This phosphorus right here has a very large partial positive charge. So serine can attack the phosphorus, and then ultimately the fluorine is kicked off as HF, and now we have what we called previously an enzyme inhibitor adduct. And an adduct basically tells you that there's a covalent bond between an enzyme and an inhibitor. It is long-lived, a very long-lived adduct. So what's the bottom line on this? If you are exposed to sarin gas, the active site of acetylcholine esterase becomes blocked. Acetylcholine can't bind. Acetylcholine accumulates, and that causes a great deal of, I'm just going to call it physiological distress. What we know is someone exposed to sarin gas will die within about 10 minutes due to suffocation associated with lung muscle paralysis. And it's not a pretty sight. In fact, if we were in class, I wouldn't even show you a video of this because it is quite gruesome. Sarin has been used as nerve agents, as chemical warfare agents in a variety of settings, and most of the people that suffer are young children. But this is an example, though, of irreversible inhibition of an enzyme. All right, so what we're going to turn to now is something that's not enzyme inhibition, not enzyme inhibition, but it's related to enzyme inhibition. In fact, I'm calling it a competing reaction. It's basically when you don't have an inhibitor of an enzyme catalyzed reaction, you have an alternative substrate. So let me set the stage for you in our first issue here. All of us have an enzyme called thymidine kinase. We need thymidine kinase because thymidine kinase converts deoxythymidine to deoxythymidine monophosphate, and then another enzyme converts that into deoxythymidine triphosphate. Why do we care about that? Because this is how we put the base thymine into DNA. And so this is a very important reaction in DNA synthesis. So we have this enzyme. Many animals have this enzyme. Viruses have this enzyme. So years ago, in an effort to treat herpes simplex virus 2, there have been some drugs produced that can actually provide an alternative product if you have a drug that acts as a substrate for thymidine kinase. So let me walk you through this. So it's time to take charge. If you have herpes simplex virus 2, typically a sexually transmitted disease, then you may be prescribed Valtrex or Zovirax, and I've shown you the structures of the two here. Actually, Valtrex is called valacyclovir, and then the two drugs are similar. In fact, if you're prescribed Valtrex and you take it, Valtrex is an example of what we've called before a prodrug. It is actually metabolized by an esterase enzyme in the liver, and then you cleave the ester bond, and then you get Zovirax, which is called acyclovir. So whether your doctor prescribes you Valtrex or Zovirax, you end up with Zovirax because Valtrex is just metabolized into Zovirax. So what do we know about this? Well, how is this going to be antiviral? How is this going to stop the propagation of the herpes simplex virus number 2? Well, here's how this works. Zovirax is a substrate. It's not a competitive inhibitor of thymidine kinase. It is an alternative substrate for thymidine kinase. And Zovirax, let's call this acyclovir, is converted into acyclovir monophosphate. But this only happens with viral thymidine kinase. Our thymidine kinase, human thymidine kinase, will not accept acyclovir as a substrate. Only viral thymidine kinase will. The bottom line is acyclovir monophosphate is ultimately converted into acyclovir triphosphate, and then you get a disruption of viral DNA synthesis. So acyclovir triphosphate basically is a poison to DNA synthesis. It will stop DNA synthesis once acyclovir triphosphate gets incorporated into the DNA. So this is a very clever strategy. Did we block viral thymidine kinase with an inhibitor? No. We feed it a substrate that will ultimately be converted into this kind of poisonous molecule just to the virus. Why doesn't it hurt us? Because our thymidine kinase will not accept acyclovir as a substrate. So again, we called this a competing reaction. It's a way to not cure someone of herpes simplex virus 2 infection, but to suppress propagation of the virus, because this will disrupt viral DNA synthesis. All right, what's another example of a competing reaction? Well, Alcohol dehydrogenase is a very important enzyme in metabolism. It metabolizes ethanol. Uh, every alcoholic beverage consumed in this country, in the world, is based on ethanol. And ethanol is metabolized by the enzyme alcohol dehydrogenase to produce an aldehyde here. So here's an aldehyde. And then further metabolism gets you to acetyl-CoA. And then you just go on into human metabolism. So ethanol really is the only alcohol you can drink in moderation without it being highly toxic, but it feeds into human metabolism, so it does have caloric value. So overconsumption of ethanol-based drinks can lead to significant weight gain. Now, alcohol dehydrogenase can also accept some other substrates, and one of them is methanol. And so what we know is alcohol dehydrogenase here. Where are you going to find methanol at? Well, you know, one of my favorite shows in the wintertime is Moonshiners, and if you watch Moonshiners, they make ethanol-based moonshine, grain alcohol. And so, but when they first start making it and the alcohol starts coming off the still, they say, well, you can't drink the first part of this because it contains methanol. And that's true. Methanol can also be found in other variety of sources. What we know about methanol is that consumption of methanol, enough of it, can make you go blind and eventually lead to death. Why is that? Because methanol can be metabolized to a molecule. Methanol is a substrate for alcohol dehydrogenase. And once alcohol dehydrogenase metabolizes it, you get the molecule formaldehyde. Well, you may have heard of formaldehyde before. In fact, one day you will meet this molecule in a very abrupt way. It is embalming fluid. Formaldehyde is toxic. So the bottom line on this is, if someone accidentally ingests methanol, one of the treatments is give that person ethanol. Is that an example of enzyme inhibition? No. It's an example of competing reaction. So the ethanol can take the place of methanol in the active site of alcohol dehydrogenase. Hopefully you can over time excrete out the ethanol. Hopefully you can over time excrete out the methanol. It's not converted to formaldehyde and it doesn't cause you a problem. So again, competing reactions, not competitive inhibition, not inhibition in any way. It's just a side reaction that has medical benefits. Another example of something alcohol dehydrogenase can do is it actually is responsible for the toxicity of ethylene glycol or antifreeze. We know, hopefully we know, ethylene glycol typically is used as an coolant or antifreeze in internal combustion engines. The problem is it is highly toxic because animals will convert ethylene glycol via metabolism of the enzyme alcohol dehydrogenase to a molecule called oxalic acid. Oxalic acid loves calcium ions, and it will form a calcium salt called calcium oxalate. This molecule will precipitate in the kidneys and can lead to significant pain and even death. Well, humans hopefully have been taught not to drink ethylene glycol, and in fact manufacturers put this really kind of green tint color right here, you can see, in antifreeze, because that will help people realize, hey, this isn't water, maybe I shouldn't drink this because it has this funny kind of green tint to it. And so we know not to drink it, but unfortunately our furry friends, especially dogs, don't realize this. Ethylene glycol has a sweet taste, so especially dogs will want to lick it up off driveways where there's been a spill of antifreeze, and it can cause significant problems, even death. If you're interested, they make They make pet-friendly antifreeze called propylene glycol that works about as well as ethylene glycol, and it will, for reasons we're not going to get into, when it's metabolized into a normal molecule that's in animal metabolism. So, hey, chapter 5, we're done. Thank you.